Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Professor Herbert Edelsbrunner from Institute of Science and Technology in Austria, IST Austria, who will talk today about the intrinsic volumes of space filling diagrams and their derivatives. Over to you, Herbert, please.
All right. Thank you, Vitaly, for inviting me. This talk kind of summarizes several papers. And so there will be a lot of details which I have to gloss over. And I try to just pick a few things that maybe highlight the general results. I also want to say this is a long-term collaboration with Patrice Koehl, Robert Bryant. Robert is at Duke University, then Michael Levitt. Michael Levitt was part of a biogeometry grant that was at the beginning of my time in Duke University, beginning of 2000. And then later, I worked with Arseny Akopian on, in particular, the derivatives of the curvatures, mean curvature and Gaussian curvature. All right. And so it's really a summary of the following 4 papers. The first 2, they were with Robert Bryant, Michael Levitt, Patrice Koehl, was about the area and the volume. And the reason was because at that time, some of the free energy was thought to be related to the area and the volume of a protein. And so that's what we focused on. And with area and volume is easy to compute, but the derivative is a little complicated. And so that was somewhat technical work. The area derivative and the volume derivative, in particular the area derivative, I remember there was that we had an error for a long time, and then it turned out it's not just a pairwise interaction, but it's a triple-wise interaction that it's relevant, but maybe I'll get to this later. All right, and then many years passed by, and then in 2020, I worked with Arseny on the weighted mean curvature and the weighted Gaussian curvature. And the reason for this is because in the meantime, I ran into Roland Roth, Austrian biophysicist who's working in Germany. And then he told me about what they call morphological approach to free energy, which says not only volume and area is important, but maybe also mean curvature and Gaussian curvature. And I kind of listened to him and it made some sense to me, but then many years passed by. But then in 2020, we did the work. And Patrice Koehl, after I talked to him, implemented the whole thing. And so there's a 5th paper, and the 5th paper is probably the one you want to read if you want to read any of them, because the 5th paper gives more of the biology background and the motivation, and it summarizes the fairly technical mathematical results. All right. Also, as you all know, giving a talk on Zoom is sometimes a little boring because I'm really talking to my computer and I only see your pictures. So if you have questions, if you would like to interrupt, I would very welcome that. And then we can have some interactions also during the talk.
We will, Herbert.
All right, very good. Okay, so this is the plan. Let's talk about the biochemical, biophysical motivation. So the motivation is mostly from molecular dynamics, where you would have some system, maybe a protein, maybe something more complicated, maybe several proteins in solution. And molecular dynamics does a simulation of the motion of the system within the solution. Now, maybe in the last few years, the question comes up, why do you do this for the proteins anyway? Because now there's AlphaFold, and AlphaFold predicts the geometry of proteins very accurately for many of them without any molecular dynamics. And I guess an answer would be that that is true. AlphaFold works mostly by pattern recognition and similarities, but there's lots of problems that it does not solve. It gives you a shape, but it doesn't give you an assembly of shape. It has problems with motion and so on. And so molecular dynamics, I think, really has something to say still. But molecular dynamics, by and large, is not solved or not consistently solved, so we can rely on it. So that's also the contribution of this morphological approach that I told you, namely trying to improve the molecular dynamics so that it's more accurate over long time sequences. All right, so let's get back here. We have this protein, say in solution. There's too many water molecules, so the computation is very heavy. Therefore, people model the water implicitly, just the impact of the water or the solution on the protein, and not explicitly as individual water molecules. And for that, they make up a potential. And on a high level, this potential consists of two things: the polar and the non-polar contribution. And I just want to talk about the non-polar contribution. And the non-polar contribution has to do with when you put the protein into solution, it needs some space. So the water molecules need to move out. And there is van der Waals interaction between the surface of the protein and the solution. And this second part, so W sub NP, the non-polar interaction, that is what I want to model with intrinsic volumes. Briefly about proteins. So a protein is roughly consists of a backbone. And from the backbone, there are side chains hanging off, amino acids. They're usually 20 different amino acids. And so it's a long sequence of amino acids. And this long sequence folds up. And the first diagram that you see here is called the cartoon diagram. I think this was invented by Jane Richardson from Duke. This gives you a global picture, so how the backbone folds and twists in three-dimensional space. If you actually draw the molecules plus the covalent bonds, then you get something called a stick diagram. That's the middle diagram. It's a lot messier because you don't see so nicely anymore how the backbone folds in space. And then the third picture is sometimes called CPK model. I call it a space filling diagram, where each atom is a sphere. The size of the sphere depends on the type of the atom. And so it's a union of spheres. And this union of spheres makes some kind of shape. All right. In the 70s, I think Leon Richards had a big contribution there. It became more or less clear that proteins actually have a shape, which is kind of amazing because they are just chemical objects. But they in solution, they fold up. And so usually the same protein has the same shape. Not entirely true, like everything in biology is not always true. So there's no theorems. There's only 99% theorems or something.
Herbert, could I ask here, when you say the same protein, do you mean the same sequence of amino acids?
Correct. Same sequence of amino acids. Yeah.
Okay, thank you.
And Eisenberg and Lachlan in the 80s came up with this idea that the non-polar part of the free energy corresponds to the area. And here I write the area sub I is the area of the ith atom. W sub I is a weight, for example, the hydrophobicity of this of the molecule. And then you just take the sum. So this is a sum of, this is really an area, but I do it weighted. So allow some physical input there. Okay, so this was what I mentioned earlier. But then there were some experiments, and the experiments showed apparently that sometimes the volume seems to be a better match for this energy than the area, in particular for small systems of molecules. Oops.
Herbert, when you write area I, do you mean exactly the surface area of a single ball, or is it more complicated?
Yes, I exactly mean that.
So it's simply 4 pi r squared.
No, because not all of it is on the surface.
So you take only that part of the surface that is not covered by other balls.
Right. So I'm talking about a fraction of the full area of the ball, of the eyeball.
Okay.
In fact, we will talk a lot about the fractions later in the talk. So as a further step after conjecturing that area or volume or a combination of the two might be responsible for it, Koenig, Roth, and Mecke in 2000 something conjectured that maybe it's all 4 intrinsic volumes. And I'll talk a little bit about the intrinsic volumes because this is a beautiful theory within mathematics. But in 3 dimensions, the intrinsic volumes are what we call the volume, surface area, mean curvature, and Gaussian curvature. And here I mean integrated over the entire surface. So the integrated or total mean curvature and integrated or total Gaussian curvature. And in case you wonder about the Gaussian curvature, because the Gaussian curvature is, the integrated Gaussian curvature is invariant as long as the topology of the surface doesn't change, Gauss-Bonnet theorem. But it is, in this context, still a difficult problem because we have these weights. Every atom is weighted and so has different contributions. So in this context, the Gaussian curvature can actually change as there's motion in the molecule. Okay, so this is the morphometric. I shouldn't have called it morphological, morphometric approach. And we work with what's called the accessible surface. So this was introduced by Lee and Richards. That's the surface and then the contributions and stuff. All right. Okay, and why intrinsic volumes? Because intrinsic volumes pop up in mathematics in many places. Therefore, they also have different names. They're sometimes called Quermaßintegrale is essentially the same, but normalized differently. And then there's also killing curvatures. And in physics, I think they're usually called Minkowski functionals. It's all the same thing. I like to call them intrinsic volumes. And really the main theorem that started this whole thing is due to Hugo Hadwiger from the 50s, last century, called the characterization theorem, which says that for convex bodies, if you have a measure which is continuous, invariant under rigid motion, and additive, then it's a linear combination of the intrinsic volumes. And so that's why the intrinsic volumes are important, because if you know them, then you know all the measures that are continuous, so stable under slight changes. You can move them in space. And additive really means that if you have 2 bodies that overlap, then the measure is the measure of A plus the measure of B minus the measure of the intersection, inclusion, exclusion. All right. So I want to say a little bit more about intrinsic volumes. So really the theory of intrinsic volumes is only for convex bodies. Proteins are highly non-convex. But this theory has been extended to non-convex in limited situations. I take some liberty and apply things to non-convex things. So mathematically, maybe sometimes it's not entirely justified, but it's motivated by what's known about the convex case. So to give you a feeling of the intrinsic volumes, I like to start by explaining the Steiner polynomial of a convex body. And here in my picture, I have my convex body is a cube. So the blue edges, that's the unit cube in space, just because it's simple and so things can be understood easier. So that's my convex body. Then I take the parallel body of the cube, which means the Minkowski sum of K and a little ball with radius r. And suppose r is small. So when you do this to the cube, then the edges get rounded off. At the vertices, you get little spheres. So that's called the parallel body. And then the theorem by Jakob Steiner is that the volume of this parallel body is a polynomial in R of degree the dimension of the space. So we are in 3 dimensions, so we have a degree 3 polynomial. So there are 4 terms, A0, A1 times R, A2 times R squared, and A3 times R cubed. And these coefficients of the polynomial are A0 is, of course, the volume, because for R equal to 0, the parallel body is just the convex body itself, so it's the same volume. A1 is the surface area, which is also intuitive because to the first order, the change when you grow the body is surface area times the radius. But then at the edges, you get little tubes. So along an edge, you get, and a cube, you get a quarter of a cylinder. And the volume of the cylinder is the length of the edge times r squared pi divided by 4, because it's a quarter. And then at the vertices, you get r cubed, because you get these sphere pieces. And I wanted to say a little bit more details about particular the A2. So this is the contribution along the edges. So that relates to mean curvature. In the unit cube case, this is 3 pi. And I wanted to work out why this is. Because we have 12 edges of length 1. All right, so that's 12. And then we multiply this with r squared pi divided by 4. 12 divided by 4 is 3 r squared. Oh, yeah, 3, yeah.
Yes.
Pi. And the r squared comes in the polynomial. So A2 is 3 pi in the case of the unit cube. All right. But in any case, so the theorem of Jakob Steiner is that no matter what the convex body is, this is always a polynomial, gives the volume exactly of the parallel body. And the coefficients of this polynomial are the 4 intrinsic volumes, or rather, sometimes you have to, it's a multiple of them. So to figure out the multiple, sometimes I use Crofton's formula. This is also a beautiful subject, less important today, but I wanted to mention it. Because Crofton's formula says that you can think of an intrinsic volume as the integral of the intersection of the body with all planes of some dimension. For example, if the planes are dimension 0, that means points, then it's just the integral, so you get the volume. When you integrate the intersection with lines, and you always take the Euler characteristic of the intersection, you get the area, but there's a coefficient here. And I calculated as 2 over pi. And the mean curvature is 1.
But Herbert, why is it area when you intersect with straight lines?
Why is it the area? Oh, well, that's Crofton's formula.
Ah, I see. So it's the formula gives the area in terms of the Euler characteristic of intersections with straight lines.
Yes, it's not so difficult to convince yourself that it's actually true. So I don't think it's plausible that it is, because you integrate over all lines in space. And then you can imagine the line intersects the body. So you get an area contribution where the line enters the body and where it leaves the body. The intersection of the line with the body is some number of intervals, and the Euler characteristic just counts these intervals. All right. But then you have to ask yourself, like, how much of a contribution do you get where the line enters? And that's because you look at all lines, so you can look at all lines when they enter. And so you get these different angles, and then you integrate over the angles, and then if you integrate everything out, that's the formula that you get.
So could you check the function under the integral is actually integer?
Is integer, yes. Yes, it's the Euler characteristic, yeah. Yeah.
So we're integrating a function with integer values, but get a real value.
Absolutely.
Okay.
You can try it out, for example, for the sphere. So for the sphere, it's relatively easy to do a back of the envelope, and then you get the volume of the sphere. But maybe what's less intuitive, so the lines and the area, I think you can make plausible. But the next step, when you take planes and intersect the plane with the body, then you get the mean curvature. That is not so plausible to me. It's true, but not as easy. Oops. Had some problems on the computer. All right, we're back on track. All right. So I wanna compute these four intrinsic volumes: the volume, area, mean curvature, and Gaussian curvature. And here, I'm developing so gory detail technical formulas, and so that you can actually compute it. And it's based on alpha shapes, fractions of spheres, and maybe some, maybe not all of these letters here I can explain. Maybe let's start with the volume. So to get the volume, I take all the spheres, so I index them by I. The Wi we can ignore because that's a physical value that's, let's say it's 1. Nu sub I is the fraction of the ball inside of the protein. I use fractions here because balls overlap, and I don't want to double count where they overlap. So I'm going to clip it with Voronoi diagrams, and so I'm talking about fractions. And then r cubed is just the how it scales with the radius. For the area, I use sigma sub I as the surface fraction of the ith sphere. So the fraction of the ith sphere that's on the surface of the protein. Let's see, for the mean curvature, we have contributions along the surface. But we also have contribution along the circles where two spheres intersect. So this is getting a little complicated, but we'll talk about it later. Here I just explain the letters. So I have sigma ij. Ij are two spheres, sphere I and sphere j. They intersect in a circle, and a fraction of that circle will show up on the boundary of the protein. And that fraction is sigma ij. Rij is the radius of the circle where the two spheres intersect. Phi ij is, if I remember right, you take a point at the intersection, you take the two normal vectors of the two spheres, it's the angle between them. Yeah, there's a few more like alpha I, alpha J, alpha K. I'll explain them later. But essentially, when we do the computations of the intrinsic volumes, we have to figure out how to find all these things. And then I'll tell you also how to make them into the derivatives. Oops, I'm going backwards.
Herbert, this formula are the main mathematical theorems.
This?
Yes, from your work, right?
No, 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 no. Well, this has nothing to do with the derivative yet. So this I
Ah.
consider easy. Maybe it could be a lemma.
Okay,
I
okay.
mean, this is just writing it down in, it might look complicated to you because you're not familiar with exactly the notation, but it's sort of an obvious thing, but it's a starting point. And so there are lots of fractions here, fractions of volume, fractions of surface, fractions of length of a circle, and others, as you will see later. And they come from the Voronoi diagram. So let me introduce this. So the basic approach to computing volume, for example, is inclusion-exclusion. So I assume you're all familiar with this. So if I have a set of n shapes, then the volume is just the plus minus inclusion-exclusion volume. So the sum of the volumes minus the sum of the volumes of the intersections, plus the triple intersections, minus the four tuple intersections, plus the five tuple intersections, and so on. So this is very good. The problem is there's exponentially many terms. In the 70s, actually, Kratky, who is an Austrian physicist, I never met him, but I tried to meet him when I was in Austria, I remember. And so I looked him up on the phone book, but apparently there's so many Kratkys. And then also I found a few Kratkys who are at the university working in chemistry and physics. And so I contacted one, and then he said, well, it's not him, but maybe somebody in Vienna. So I never quite figured out who that was. Anyway, so Kratky proved that if you have a union of spheres, then the inclusion-exclusion formula can be cut, shortened, so that you look only at terms up to 4 spheres. So it's the sum of the spheres minus the pairwise intersections plus the triple intersections minus the 4 tuple intersections. That's enough.
Herbert, this is for balls in three-dimensional space.
This is for balls in three-dimensional space, yes.
How about high dimensions?
Balls in higher dimensions, you get up to d plus 1. D plus 1 balls.
And in the plane, is the plane analog?
Only to 3. So disks minus the pairwise intersections plus the triple-wise intersections.
And in the three-dimensional space up to quadruple intersections.
Yes. But I should mention that if you take all the pairwise intersections, all the triple-wise intersections in a plane, for example, this is not correct. Kratky only proved that there exists a selection of spheres, pairs, triples, and quadruples, so that the inclusion-exclusion formula is correct. But he didn't tell you which one it is. But somehow from the whole equations, that's all. You can eliminate and then find out a formula like that. But I'm going to do a shortcut. So I'm going to tell you straight away what the formula is going to be. The only thing I need here is I need the notion of a Voronoi diagram. So here is my drawings are two-dimensional. Here is a union of disks. I'm also draw the Voronoi diagram of the union of disks. And it decomposes the union into convex pieces. And now, for example, I can define nu sub I. I told you this is the volume fraction. So I take a disk and only a fraction of that disk is actually inside of its Voronoi region, and that's nu sub I. Nu sub ij would be the fraction of the disk in 3 dimensions where 2 spheres intersect. That's in its Voronoi tessellation.
Herbert, this is probably some weighted case.
Oh yeah, in the picture it's a weighted case, yes. So instead of the Voronoi, I take a weighted Voronoi decomposition, which is sometimes also known as power diagram, or I think Laguerre diagram sometimes. Yeah, because you see it you see my cursor, right? Yeah, for example, this is a smaller sphere and this is a bigger sphere. And in order for the bisector to go exactly into where the two surfaces intersect, it must be shifted a little bit. And the power diagram does exactly that. But really the geometry of the power diagram and the Voronoi diagram is almost indistinguishable. So usually when I talk to biologists, then at the end of the talk, they always say, and what if the radii are different? And then usually I say, well, everything goes through. And so here I draw a picture where the radii are a little different so that, yeah, so it's essentially no difference in if there are different radii. But instead of Voronoi, you used power diagrams. Okay, so if I now decompose my protein with a Voronoi or power diagram, and then I take the nerve, which is kind of the dual graph of it, where I connect two centers of spheres if they share this a common face. In 3 dimensions, this would be a sphere or a piece of a sphere. Then I have triangles. If a triple has an intersection that I have in 3 dimensions, I have a tetrahedron. If 4 balls in their Voronoi decomposition share a point, this is also known as the alpha complex, this dual structure. And this alpha complex is very useful because what I told you about the inclusion-exclusion formula, if you now take a term of the inclusion-exclusion formula for each simplex in the alpha complex, then you get a correct formula. So this is the theorem. I proved it in 1995. So what this says is the volume is the sum over the volume of all balls minus intersections of balls, where I only take the intersections that correspond to edges in the alpha complex, plus triples, where I only take the ones that correspond to triangles, minus quadruples, which I take the ones that correspond to tetrahedra. I think I do something that sometimes I cannot go back and forth. Okay. And then so that you get a feeling of how these things look. So a ball at the intersection of 2 balls, intersection of 3 balls, intersection of 4 balls. So when you use this formula, you only need to compute the volumes of these things. Maybe you think I cheat a little bit because, for example, the triple intersection of 3 balls does not always combinatorially look like that. In particular, if the balls are different sizes. But it turns out that if you take the triples that correspond to the alpha-shaped triangles, they all look like that. So combinatorially, it's always the same type. That simplifies the implementation tremendously because you always have only one case. And then also the 4-tuple intersection, combinatorially, it really looks like a tetrahedron, but it's the intersection of 4 balls. Similar formulas that from the one that I just told you about alpha shapes also exist for surface area, length. So for all the parameters that I showed you, they can be done with inclusion-exclusion. And it's always a little bit different. I just want to explain the one super volume. All right. Actually, I'm a little fast, so maybe you want to ask some questions. Is everything clear so far?
I feel that this theorem on using alpha complexes is actually very strong.
Oh, this theorem?
This theorem.
Yeah, it's beautiful. I'm very proud of it. I mean, there have been developments a little bit in this direction. So the first step was what I mentioned by Kratky, where he proved that there exists something like that. But he didn't say what. And then there's also an interesting story about it, because Kratky's idea was then implemented by Sharaga, who is a prominent biochemist from Cornell University. But this reduction, where you remove terms and replace them by more but shorter terms, has some problems, and there was a bug in the software. And this bug, I mean, the software was very popular, so everybody had the same bug. So they could compare their results, although it was a bug. Anyway, so we implemented my formula. It didn't have the bug, so we get different volumes. All right. So then I was working with a biochemist in Urbana-Champaign. So this was a long time ago. And then so she used our software to compute all kinds of things like holes, surface area, volume, and so on in order to study proteins. But then she needed to compare her results with other people's results. And the trouble was that other people's results were done with a wrong formula. And they were all using the same wrong formula. So then she felt compelled to use also the wrong formula so that it's comparable with other results, which I thought is a interesting but very strange scientific move, that you actually use something wrong because the majority of the people do that. Anyway, so science is kind of curious sometimes.
So it was published in the wrong way when.
Well, people knew that sometimes there's a mistake. The mistake is not huge, so people didn't really care that much. But then if you compare in details to other people's results, if you have the same bug, then it's comparable. Then it's easier to compare.
Herbert, may I ask if this formula holds in the periodic case?
Periodic case? It always holds.
So when we have our atoms in the periodic crystal and then compute
well, the volume is infinite, right? So
yeah, of course we compute it within a unit cell.
Oh, within a unit cell. Yeah, you can do that.
So I think that it simply works straightforwardly.
Yeah.
Okay.
Yeah.
Great. Thank you.
Yeah, and then maybe I could also mention, so there was one step in between after Kratky. Daniel Neyman and Henry Wynne, they proved a similar theorem to the one that's on the slide right now. But instead of the alpha shape, they put the Delaunay triangulation, the entire Delaunay triangulation there. So this is also true. It's also a theorem. But the Delaunay triangulation, of course, has more terms. Many of those extra terms are zero, but there can also be extra terms that are not zero. And then somehow it turns out they all vanish. So the alpha shape formula is preferable because it doesn't have any redundant terms. But a big advantage, in particular in implementation, is that when you use the Delaunay triangulation, it's not true anymore that there's only one case for each number of intersections. Then once of a sudden you have combinatorially different types of 4-tuple intersections and 3-tuple intersections. And it's a little painful because then you have many cases. But this theorem with the Delaunay triangulation also, this holds in all dimensions. So actually everything I said here holds in all dimensions. In every dimension is just one up. All right. Let's get to the formulas. So to give you a feeling of what the formula is, so this is the theorem about the area derivative. So how do I think of it, first of all? I think of a a protein as n spheres or balls in 3 dimensions. And if all the radii are the same, then I can think of this as a point in 3 n-dimensional space. I just concatenate the coordinates of the centers. Even if the radii are different, I think of it as a 3 n-dimensional point because the radii don't change. At least what I do is I look at the derivative if each atom moves in a certain direction. All right, so X is a point in 3 n dimensions, so that's a protein. T is a vector in 3 n dimensions, and so T gives you a little vector for each atom, how it moves. Okay, and for a given T, I would like to give you the derivative. And I give you the derivative as the scalar product of a vector A, and I'm going to tell you what A is. So A consists of all the measurements that I talked about or skipped over, and multiplied a scalar product with T. This is the momentum vector. Okay, so this is a compact way of saying it, but really it's for each atom, I have the momentum vector of the atom, and then I have some 3-dimensional vector, and I take the scalar product, and so this gives the contribution of this one atom to the derivative. So everything is decomposable. And so here is the recipe of how you get this vector A in 3 n-dimensional space. It's a while ago, so I forgot what those things all are. So A, let's see, A sub ij is here. What do we not know? So xi is the center of the ith sphere. uij. Uij is the vector from the center of the jth sphere to the ith sphere, unit vector. Uijk is the unit vector from the ith sphere normal to the vector between j and k. So it's geometric entities like that. And the rest of it is a lot of analytic geometry. So this is Aij. Let's see, the radii are here. Centers are here. Oh, here is Rijk. Let's see, Ri was the radius of the sphere. Rij was the radius of the disk or the circle in which 2 spheres intersect. If you intersect 3 spheres, you get the 0 sphere, which means 2 points. And it's the radius of this 0 sphere. So it's half the distance between these 2 points. So that's Rijk. And nu ijk is, so when you decompose this thing with a Voronoi diagram, then you look at 3 spheres that intersect. So you have this 0 sphere between the 2 points where they intersect. These 2 points might be on the Voronoi edge of the 3 spheres. Then nu ijk is 1. But maybe only a fraction of this edge between the 2 points is inside of the Voronoi diagram. And then this fraction is nu ijk. Okay, I try to be consistent with these fractions and the notation. All right.
Herbert, if you swap 2 points, so the action of the permutation group respects all this formula, right?
Can you say it again? I couldn't follow.
So if you swap indices of 2 atoms, right? When we similarly swap indices in all this formula.
Yes.
Right. So the order here is not really important.
The order is not important. That's right.
Mm-hmm. Thank you.
Okay, here are a little bit of explanation. So as I said, Uij is this vector between the ith center and the jth center, normalized to unit length. Uijk is visualized down here. Oh, the definition is different from what I said. It's the unit vector from the kth center normal to the line between the ith and the jth center. Oh, wait, it's not the unit, but it's rather the projection of the unit vector from I to k onto this direction.
Herbert, in the second formula, is it possible you divide by the length of vector in the numerator, right?
Yeah, it's unit length. Sorry, I misspoke. It's unit length. Yeah.
Okay.
Yeah, this just means I divide by the length of the denominator so that you get unit length.
Is it possible that this vector is simply 0?
No, because if it's 0, the three spheres don't intersect in a 0 sphere in 2 points because the centers are parallel. And then if the centers are parallel, this triangle cannot be in a Delaunay triangulation or in alpha shape. So you will not get such a triangle.
Okay.
Yeah. Yeah, fortunately, lots of these kind of nasty cases are eliminated by the construction. I don't think I can make it justice, but this term Uijk, is very interesting because I couldn't explain it anymore. But for a long time, I thought the area derivative should be only sensitive to pairs. So any two pairs, whether they get further or closer. But that's actually not true. So it's triple intersections when two gets closer, how a third sphere interacts with them is also important. But I think I talked myself into some contradiction when I tried to explain this. But I thought this is very interesting, and eventually we got it right. And I want to say a little bit about the Gaussian curvature, just because there's new things showing up. For example, one thing we have to struggle with in mean curvature and also in Gaussian curvature is there's contributions along the circles of intersection. And in Gaussian curvature, there's also a contribution at the vertex. And since the spheres have different weights, we have to decide of how much contribution of the curvature along a circle, for example, goes to the ith and to the jth sphere. And we decided half and half. And the justification for that is if you think of the union of spheres, which is not a smooth surface because the intersection, and then you have a sharp crease at the intersection. You can approximate it by rolling a ball along it, like to get a little torus piece. So you get a round edge. And then you let this sphere that rolls along it go to 0. And then you take the limit. And if you do that, then it turns out that if you divide up the volumes accordingly, then along a circle, the contributions are equal. So here we have the weight of I plus the weight of j divided by 2. So this is along a circle, but along at the vertex, in a Gaussian curvature case, it's more complicated than that. And there we have to introduce alpha I, alpha j, and alpha k. These are the fractions of contributions at a vertex, which depends on the geometry. And I want to explain how it depends on it, but I cannot, explaining why is a little bit too much, I think. But how I can. So now I'm talking about three spheres. They intersect at a point. And this point is vertex on the boundary of the protein, of the union of spheres. So at this point, I have 3 normal vectors, one for each sphere. I call them Ni, Nj, Nk. So these 3 normal vectors that define a solid angle, which is drawn here. And it turns out the right way to partition the contributions to the things is you take this spherical triangle, you divide it, so Z is the center of the circumcircle of Ni, Nj, Nk, and you connect Z to the midpoints of the 3 edges. All this happens on the surface of a sphere. And then alpha I, that's the fraction that's allotted to the ith sphere, is the fraction of the area of this quadrangle, spherical quadrangle, over the area of this of a spherical triangle. So this is alpha I, alpha j, alpha k. And then maybe you ask now, can alpha I be negative? And the answer is yes, can be negative. But the geometric configuration where this becomes negative is a little strange. But still, to have everything consistent, this is the way to go. All right. Got to the last slide. Second to the last slide. So we have these theorems. I just showed you 2 of them, but we have all 4 of them and all kinds of details. But it turns out that the derivative sometimes is infinity. It's infinite or not defined. And one advantage of the formulas that we found is that I can tell you exactly when the derivative is not defined, and it corresponds to geometric configurations that are not generic. So typically when 2 spheres touch at a point, like drawn here, when 3 spheres pass through the same circle, I think when 4 spheres pass through the same 2 points, and maybe that's it. And so we have a classification of when the derivative is infinite, and it's not defined mathematically. I mean, there's a clear reason why it shouldn't be defined. But if you do it, and this is now a comment of other approaches, if you do it numerically, because I mean this problem has been solved numerically to some extent, then you get something not defined. You never know why. Maybe it's a machine error, maybe some this or maybe that. Anyway, so this you can tell here. And my last slide is thank you for listening.
Thank you very much, Herbert. Please thank Herbert for the nice presentation, please. Yeah, physically, virtually. And yeah, we have several minutes for more questions. But usually at that point, I stop the recording.